So thanks again for your time today for joining our webinar at Sargentis. Again, good morning, good afternoon, or good evening, based on where you are. My name is Alexander Jensma, and I'm the Sales and Account Manager for the application field of cell and gene therapy at Sargentis, and I'll be hosting today's webinar. So the webinar's title of today is the next generation sequencing-based characterization of transgene insertion for manufacturing of viral vector producer cell lines and immunotherapy products, with a specific focus on cell and gene therapy. I'll be giving a broad overview of what TLA is, what its technical background is, what the different applications are for its use, the output we generate, and then specifically what services we offer in the fields of cell and gene therapy. And I'll provide some specific hands-on examples from publications in the past that made use of TLA. So as you may know, there are several challenges in cell and gene therapy characterization and approval and clinical practice at the moment. For example, when a transgene is used, whether the integration occurred at the intended locus, if the integrity of the vector remained intact, and in viral integration types of processes, whether there is a dominant clone, a dominant integration site, and for example, in the CAR T space, whether an integration occurred in an oncogene or tumor suppressor gene. But also more practically speaking, what the efficiency and accuracy of the method for targeted integration was, and perhaps to evaluate and compare different methods, but also what the cost effectiveness and the turnaround times are of the different methods currently being used. In general, there's a need for in-depth genetic quality control. As you can see, there are many different reasons for quality control in cell and gene therapy at the moment. For example, the assessment of stability and genotoxicity in ATMP types of studies, and the long-term monitoring of safety and the clonality of cell therapy products, especially in patients. Here you can see a broad overview of the cell and gene therapy process. There are three main areas of interest where we can help as Sargentis using our target locus amplification technology. So this is firstly in the viral vector producer cell line, which can be directly applied in the field of gene therapy, but the vectors which can also be, of course, used for downstream transduction of CAR T or CAR NK or other types of modalities for final use in patients. And of note is that TLA has been extensively published in literature for all of the different methods used for the editing and transduction of both gene and cell therapy, as you can see here. So random integrations, as well as knockouts and homology-directed integrations or targeted integrations. All of these publications are freely available on our website. As you may know, there are different conventional approaches to determining genetic variation, such as whole genome sequencing, PCR, ddPCR for determining the copy number, southern blot, and FISH. So you may know that FISH is the primary method traditionally for characterizing integration sites, and PCR for determining small variations and mutations, and whole genome sequencing as a more generalistic approach for determining the DNA sequence content inside of cells. And with this comparison in mind, TLA in combination with next generation sequencing, so as I will explain later, we make use of Illumina short read sequencing. TLA allows you to capture all of the genetic variations listed here, so integration site, but also structural variations, co-integrations, sequence variants, and for example, copy number. And of note here that there are also primary differences with LAM PCR, as TLA allows for a complete characterization of the transgene at any location in the genome. TLA has been extensively published. We have publications in our 4 focus areas of cell line development, cell and gene therapy, but also transgenic animal models, and in genome editing, which is a more recent development in techniques such as CRISPR. We're active in 39 countries. We meet regulatory expectations, and we have a dedicated team of PhD level scientists in the lab, as well as in our reporting team, as well as in our bioinformatics team. And we currently support all of the top 20 pharma and biotech companies worldwide. So to shortly explain the principles of TLA, the TLA method is based on 2 primary principles, the first one of which is chromosome capture. So what chromosome capture means is that the 3D conformation of DNA within the nucleus of cells is captured or cross-linked, as you can see in the top left. The TLA protocol then enriches all of the nucleotides that have occurred in the same genomic locus due to the 3D conformation that we captured in the first step. And then by using inverse PCR, we generate a complete integration profile in a hypothesis-free fashion. And the result of the TLA sequencing data is the characteristic coverage profile you see in the bottom right picture, which has a significantly higher coverage than traditional sequencing methods such as whole genome sequencing, and also specifically generates a very high quality coverage profile on the transgene. TLA allows you to characterize the transgene or your genetically edited locus of interest on the single base pair level, and also sequences outward into the genome of the relative to the integration site up to 100 kb in each direction. And this information allows us to determine the integration site by mapping back the TLA sequencing data to the reference genome. And of note here is that our TLA experiments have been consistently used in follow-up experiments, for example, for characterizing specific integration sites. The coverage for TLA is very high across the vector sequence, as the TLA templates that we generate in our protocol have a very high density of the vector sequence, which are subsequently amplified using the inverse PCR reaction. But also have a very large coverage in the surrounding genomic region, allowing you to capture that information as well. As we design our primers and our experiment specifically for the vector sequence of interest, we can detect both single as well as multiple integration sites by mapping the data back against the reference sequence. So what you see here is not a whole genome coverage plot, but a TLA coverage plot to illustrate where the transgene has integrated into the genome. And why is our information used by all of the top pharma and biotech companies? And that is mainly because there are different regulatory expectations in the use of transgenes and genetic editing at the moment, which are increasingly important. And these are mainly associated with the unexpected integrations and structural changes found in the edited host genomes. So these are 2 examples published by Genentech and the Jackson Laboratory in mice, where TLA was used to discover large structural variations, insertional mutagenesis, that significantly affected the downstream, let's say, development of these products or these mouse lines. TLA makes use of 2 primer pairs, as you can see in the animation below. We do this to generate 2 independent primer data sets, and this allows us for an internal validation of the experiment. So we actually don't need a controlled sample. However, this may increase the resolution of detection in very heterogeneous samples. And as we make use of two primer pairs specifically for the vector sequence of interest, this means that we can also detect the presence of partial integrations and identify any genetic variants, as the resulting data serves as an internal control. And of note here is that for very large vectors such as BECs, we can design additional primer pairs to ensure coverage across the entire sequence. Specifically for the fact that we make use of two primer pairs, we also detect partial deletions, partial integrations, but also rearrangements such as translocations and fusions, which we provide in a tabular as well as in a visual format with the corresponding genomic locations in the host genome in the TLA report generated for each sample. Again, this is another example. TLA as such allows you to detect any presence of fusion partners as we make use of the generation of two independent data sets. So a vector may be present in one chromosome or locus, but then can show to be continuing its coverage and as such its presence in another chromosomal location within the genome. That is further visualized here. So this is an actual example of a translocation from chromosome 18 to 21 from chromosome 12, the intended locus, where primer pair 1 shows a clear translocation from chromosome 12 to 18, and primer pair 2 shows a translocation from chromosome 12 to chromosome 18. Furthermore, TLA is also used for detecting any genetic editing events, which may occur in a fashion that there is an excision and reintegration of a specific fragment during the genetic editing experiment. And this shows up in both primer sets coverage as a deletion, which I will also get back to later. And as we can then design a new experiment based on the deleted fragment that was supposed to be present, we design a third primer pair to be able to detect where and as such also how many times the excised fragment integrated into the host genome. So this slide gives you an overview of the different large structural variations we can detect using TLA, also with their visual cue they provide using the bell-shaped TLA coverage. And here you see listed inversions, deletions, which I just explained, translocations, which I explained earlier, and also a duplication event. And again, all of these events will be quantified and listed within the report for each sample. And these structural variants are further validated by the obtained breakpoint reads as well. TLA, the data generated from TLA can detect both frequent as well as infrequent sequence variants, so base pair mutations or point mutations, of which two examples are shown here. So on the left side, on the left panel, you see a vector sequence where the intended integration has occurred in all of the cells, where there was a point mutation found with respect to the reference genome. But also in a subset of reads, we can identify point mutations as shown in the right panel, where approximately 20% of the reads were shown to have the G to A mutation. And again, this is visualized within our report. So per region of interest, per sample, and per primer set, We provide the reference base pair and the corresponding found mutation, but also the coverage between the two primer sets used for the sample to further characterize these found mutations. And as you see here, we can also identify backbone sequences within transduction experiments. So in summary, what I just showed you, TLA outputs all of the genetic characteristics of your genetic engineering work, so structural variants, single nucleotide variants, but also larger variations such as concatenamers and breakpoint sequences, as well as any off-target integrations that may have occurred. Next to our standard TLA as a service, we provide different analysis packages, one of which is the clonality assurance package, which includes the characterization of a master cell bank and its divergent clone, where we make use of extended sequencing and where we can analyze up to 93 subclones and 3 controls, and where we provide 2 breakpoint qPCR analyses per sample. And this is delivered with an extensive report on your master cell bank and the corresponding clones. Secondly, we have the genetic stability package, which allows you to characterize the stability and integrity of the product of interest. So in this case, a cell therapy product or a viral vector producer cell line, for example, from the master cell bank to the working cell bank to the end of product stage. And here we make use of the different methods we touched upon earlier in this presentation. So the identification of integration sites, vector-vector fusions, and vector sequence variants. And we provide this data for all of the different steps in the production process or the manufacturing process to give a confirmation of the genetic stability. To summarize the genetic stability assessments using TLA, this provides complete genetic analysis and documentation of the genetic makeup and genetic stability across multiple generations. And from experience, we know that TLA data has also been used in regulatory filings, for example, in IND or BLA types of applications. So to touch upon some specific examples on the use of TLA in both cell and gene therapy. As I mentioned earlier, there has been extensive publication in both these fields, which you are free to explore on our website. Here are 2 examples of publications by the FDA, NIH, and GSK respectively that use TLA to characterize a vector-producing cell line. So for the example for GSK, a rapid lentiviral vector producer cell line was generated. And in the example of the FDA, a specific antigen encoding sequence in hex cells that was altered using a CRISPR strategy was further validated. But also closer to home in the UMC Utrecht, as we are located on the Utrecht Science Park, TLA was used to identify 9 different integration sites of a transgene in a viral vector producer cell line master cell bank, which again you can see here on the TLA coverage plot, which results from the TLA data being mapped back to the reference genome, and also the TLA report for this, let's say, sample would allow you to zoom in on each of the respective integration sites. As for example shown here, we provide a table with the sequence variants found in the different positions with the corresponding reference and mutated base pairs found for each of the different samples. So in this case, both the plasmid, the master cell bank, and the drug product were all characterized in the same experiment or the same project. and as such allowed the research group to compare the level of mutations between these 3 samples. TLA can actually detect a very large number of integrations. Main difference with other types of transgene characterization methods is that TLA is not limited to a specific genomic locus, but can detect any integration site within a sample based on a single experiment. So in this HIV experiment, we identified more than 1,400 unique integration sites, which is a figure used in the original publication describing TLA and its methods, published in Nature Biotechnology in 2014, which can also be found on our website. In cell therapy, TLA has applications as well. So as you may know, CAR-T and related modalities are currently a very large growing field of interest. An example of this is a paper by the Memorial Sloan Kettering Cancer Center from 2017, where a CAR construct was targeted into the TRAC locus for expression on the T cells, and TLA was in a first experiment used to identify an integration of the CAR construct in the correct locus at chromosome 14. And in a second step, the researchers wanted to further determine the level of integration of their construct. So we designed an additional experiment by placing the inverse primers at the genomic locus that we found in the first experiment. And this allowed us to identify that 15% of the integrations had a perfect homologous recombination, 5% had an imperfect homologous recombination, and up to 80% were wild type alleles with no integration event. And more recently, in April of this year, the Nationwide Children's Hospital published a study on the use of a CAR construct in NK cells, where they used the CRISPR AAV method of transduction, where again we showed an integration of the vector into the intended locus at chromosome 19, but also a low level of random integrations and no dominant secondary off-target integration sites. But we did identify a single sequence variant within the vector itself. And another example by Selectis, who is also developing CAR T cells, who made use of a double construct, so a CAR T as well as a PD construct that were transducted in the same experiment. And we designed 2 individual experiments for both vectors. And our experiment showed that the constructs integrated into the intended locus, but both with a small translocation to the other locus of interest as shown in this picture. So that concludes the technical overview of our services and the examples. So a bit about our services. We can receive both cells as well as tissue, clonal or heterogeneous samples. We have a cross-linking kit, so the first step of the TLA protocol, let's say, as we can only receive BSL level 1 samples at the moment. Which we can ship to you within 2 working days. Next to our TLA services, we offer additional services such as vector sequencing and amplicon sequencing, as well as primer design and the packages discussed earlier during this presentation. And if of interest, we can always design an agreement with you, custom reporting, meeting regulatory expectations. We are currently in the process of obtaining our ISO 17025 accreditation, and the standard turnaround time of a TLA analysis is 8 weeks and expedited 4 weeks. The TLA analysis per sample includes everything from sample pre-processing up to library preparation, next generation sequencing, bioinformatics analysis and the reporting, as well as a post-report tel call to discuss the results of the report, which is all included in the price per sample. We require 2 vials of 5 to 10 million viably frozen cells. So these can be from any source. We have isolation protocols available if needed. And as we are located in Utrecht in the Netherlands, these are where the samples should be shipped to. And we always require, in the case of transgene integrations, complete vector sequence information, which can be safely uploaded to our portal, which allows us to design the TLA experiment. We have a sample pickup and shipment service. This allows you to outsource any customs or regulations reporting for shipping your sample back to the Netherlands at a fixed price of 1,400 euros outside of Europe and 900 euros within Europe, for which we partnered with QuickStat's cold chain service. Recently, we joined the National Institute of Standards and Technology from the US Department of Commerce, which is a body that characterizes genome editing tools and quality measures, and they will evaluate and determine whether TLA is a genetic quality control tool that can be used by the different research labs and companies working in genome editing in general. So last but not least, Surgentis was founded in 2012 in the Utrecht in the Netherlands as a spin-off from the Ubrecht Institute for Developmental Biology. And we are located on the Utrecht Science Park, from where you can see a picture on the bottom right. We are currently also in the process of developing kits. So these are mostly used for cell lines and allow you to perform TLA, the TLA steps up to data analysis in-house, hereby reducing your costs. Thank you for your attention. I'll be happy to take any questions.